I'd like to welcome you to the webinar. Thank you very much for joining us. I think there will be some more people joining, but because we're tight on time, so we just get started. My name is Michael Plues. I'm a professor in developmental psychology at Queen Mary University of London. I'm one of the co-Is on the BIOPATH study, and I will be chairing this webinar today. So this is the first webinar that we are running, and we hope to be able to run several more of these. So our plan is to have one in the spring and one in the autumn, where we are able to present the most recent research findings that resulted from our research. So I'd like to get started by talking you through the program, and then I will talk a little bit about the background of the Syrian crisis, and then I'll share more about the research design of the BIOPATH study. So just sharing my slides. So I'd like to start with the program. So we'll have a few minutes where I will share about the webinar and the BIOPATH study. And then this will be followed by 3 presentations on empirical findings that are about to be published or have been published already recently. So the first presentation is from Dr. Fiona McEwen with the help of Dr. Claudinea Biazzoli on the prevalence, comorbidity, and the predictors of common mental health problems. And that will be followed by a presentation by Mrs. Cassandra Popham on the predictors of risk and resilience among Syrian refugee children. This paper has been published in summer this year, I think. And then the last presentation on empirical findings is by Dr. Demarcus Smith on war exposure, post-traumatic stress symptoms, and hair cortisol in Syrian refugee children. And then we have the privilege to hear from Professor Eli Karam, one of the co-PIs of the study. He will present some points that are important to mention and also reflect on some of the presentations. And then we have some time for Q&A, final comments, and we'll end at 3 p.m. here in the UK. Might be different time wherever you are. So we have some limited time for questions. I recommend that any questions that you have, you put them into the Q&A. So rather than using the chat function, use the Q&A function and enter your questions there. And once the presentation has ended, the person presenting will try to answer your question there, but then we'll have some time, hopefully at the end, to discuss some of the questions that you may have. So let me get started with just giving a little bit of background on the context where we collected our data. So we are conducting research on Syrian refugee children and adolescents, as well as their families. So the Syrian civil war has been going on for quite a while now. It began in 2011, related to more general unrest across the Middle East in 2011. And then initially that was a conflict between the Syrian government and several rebel groups, which then over time developed into a very complex proxy war that involved many different parties in the Middle East, on West and East. And that had detrimental consequences, which we all know. So there's about 500,000 people that died in that war. The detailed or specific numbers are not available. That's sort of the best estimate. It's probably going to be around that. It led to a huge wave of refugees. So we had 6.7 million Syrian refugees displaced internally within Syria, and then another 6.6 million Syrians that were displaced externally. And most of those externally displaced refugees actually remain in the Middle East. And about half of them are children. And we know that they're at risk of becoming ill, abused, exploited. Many of them don't have access to school. This is one of the reasons why we wanted to focus our study on these children. So we've run the BIOPATH study, which stands for Biological Pathways of Risk and Resilience in Syrian Refugee Children based in Lebanon. This is a collaboration between Queen Mary University of London, IDRAC, and the fieldwork was conducted by Mezzo du Monde. The study was funded by NICHD. So we do know that millions of children are affected by war and displacement. Unprecedented numbers at the moment, with a milestone reached of 100 million people that are displaced, forcibly displaced, many of them children. What we do know about these children is that many of them develop psychological problems, but also some of them show remarkable resilience. And we were particularly interested in those individual differences. We wanted to investigate the biological underpinnings and mechanisms that are associated with individual differences in response to war and displacement. We do that in several ways, or there's several components of our study that are important to mention. So we considered multiple settings, from the family to the school to community, neighborhood, because all those settings are important to consider, not just the family environment. We also looked at multiple levels of analysis. So we looked at the environment, but also more the social and psychological, and then down to the biological, considering neuroendocrine, epigenetic, and genetic factors. We also thought it was important to focus on both risk and protective factors, not just the risk factors, and to consider adaptive outcomes rather than just maladaptive outcomes. And most importantly, this is a longitudinal study, which is a very difficult thing to do in a refugee population that tend to be quite mobile. So a little bit about the study design. So we were interested in figuring out how war exposure and displacement was associated with both maladaptive and adaptive outcomes in these children, also considering other environmental factors and genetic factors and how they moderate and influence the effect that war exposure and displacement has on these outcomes. And importantly, we also want to test to what degree all those different experiences are mediated through DNA methylation, through an epigenetic mechanism in the prediction of these outcomes. So we collected all this data at baseline, and then a year later, we repeated everything in our 12 months follow-up. We managed to recruit a sample of 1,600 Syrian refugee children plus their primary caregivers with a wide age range from 8 to 16 years. And then that was collected in 2017 and 2018. And 1 year later, we ran the follow-up analysis data collection, which included 1,009 children and their caregivers. Just a few slides, or just one slide actually, about the measures. So we used a wide range of questionnaires to capture psychopathology, but also well-being outcomes, and then protective and risk factors at different levels. So at the individual level, social and environmental level, we tried to consider those protective and risk factors. We also collected biological samples, all non-invasive. So that was saliva for molecular variables in more detail. So we looked at genome-wide data, epigenome-wide data, so this DNA methylation. And we also collected hair samples to look at neuroendocrine variables, cortisol being the central one. The location, we did collect all the data in Lebanon, which you can see here. So if we zoom in, you can see that Lebanon is sharing a lot of the border with Syria, which explains why many of the Syrian refugees fled to Lebanon. Within Lebanon, we collected data in the Bekaa Valley, so in this area here. And I just have a few pictures to show you the context. So this is one of the 77 settlements where we collected data. These were all informal refugee settlements. We collected all the data in interviews. So here we have one of the interviewers interviewing the child. All the data is being entered into a tablet, uploaded the same day. And at the same time as one interviewer was talking to a child, someone else was talking to the mother. So as shown here, outside of the tent. And here we have a photo of all the team at the baseline data collection. So that's the short introduction. And now we'll have the privilege to hear from Fiona about the findings on the prevalence of mental health problems in this sample. So feel free, Fiona, to share your slides.
Thank you very much. Just check, hopefully let me know if you can't see those slides for any reason. So yeah, as Michael mentioned, I'm going to talk about the prevalence, comorbidity and predictors of mental disorders in refugee children. And this work has been done jointly with my colleague Cloud and I. So I will briefly outline previous evidence. I'll then talk about how we have tried to calculate new and improved estimates of common mental disorders in Syrian refugee children in Lebanon. I'll talk about the comorbidity between common mental disorders and also the multi-level predictors for symptom burden. So in terms of previous evidence, there's two recent meta-analyses that have looked at the prevalence of mental health problems in refugee children and one that looked at predictors. From these, the prevalence estimates for PTSD have been estimated around between sort of 23 and 52%. For anxiety, that's around 16 to 32%. For depression, about 14 to 28%. And for externalizing behavior problems, there's less evidence, but that is, sort of levels of oppositional defiant disorder of around 1.7% and ADHD of 8.6%. Risk factors that have come out consistently across studies include exposure to trauma, especially cumulative exposure to trauma, daily stressors, family separation, parent mental health problems, low socioeconomic status and poverty and bullying. But there's a number of sort of gaps in the literature. So much of the data comes from refugees who are resettled in high income countries, and there's not, relatively speaking, not very much data from camps and informal settlements in low and middle income countries. There's a lot of variability in estimates between studies, and this may well relate to sort of large methodological differences. Many studies have used cross-sectional designs. As Michael mentioned, it's very hard to get longitudinal data in refugee populations that tend to be quite mobile, and also sometimes non-probabilistic sampling, so there may be some sort of systematic biases in the types of samples included. There's also the risk of transcultural measurement errors, because often we take measurement tools that have been validated in one setting and use them in a different country, in a different context, and we don't always know how valid those measurement tools are. And also a sort of reliance on screening tools, and there's much less data from clinical interviews. So what we've tried to do in BIOPATH, these are some of the steps we've taken to try and get high quality data. So we've used settlement-based probabilistic sampling. So Michael mentioned we went to 77 different settlements that were selected to include a range of different levels of vulnerability. And in those settlements, we approached all families who were eligible and invited them to take part. We have longitudinal data, so a baseline and then 1 year follow-up. We used, in a subsample, we used a culturally adapted structured clinical interview. So we used the mini-KID. So the mini-KID interview was carried out, but we then had all cases discussed in clinical supervision with an experienced clinical psychologist, and diagnoses were agreed by consensus discussion after clinical supervision. And one of the aims of that was to take into account the contextual, culture and linguistic factors that might impact the diagnostic process. So we really wanted to get a sense of whether children's problems went beyond what we would see for other children in that context, and that there were problems that were causing significant levels of functional impairment or distress. So we were trying to avoid sort of over-diagnosing by sort of picking up children with sort of milder problems that could be attributed to, to their circumstances. So we only included children who had a clinical global impression severity score of 4 or greater to pick up those sort of more moderate to severe cases. And we also developed adapted screening tools, so they were locally validated screening tools that we used in the whole sample. So the measurement tools that we used in the whole sample included a number of screening tools for children's mental health symptoms. So this was the SES-DC for depression, the SCARED for anxiety, the CPSS for PTSD, and those were all self-report measures. And externalizing behavior problems are measured using the SDQ, the Strengths and Difficulties Questionnaire, and then we used additional items for conduct disorder and oppositional defiant disorder, and that was caregiver report. We measured risk factors, a number of different risk factors that I'm going to describe in these analyses. So war events were measured using the War Events Questionnaire. We measured daily stressors using a new measure called the Perceived Refugee Environment Index. We measured child maltreatment using the ICAST. Conflict between caregiver and child was measured using the parent-adolescent conflict scale, and we measured caregiver mental health through a range of validated mental health screening tools. We then calculated point prevalence estimate of common mental disorders. So we derived cohort-specific cutoffs on the screening tools that we used. And the way we did that was using that subsample with clinical interview data. And we used ROC curve analysis to estimate the optimal cutoff for this particular cohort. And that's done by comparing the screening tool to the clinical interview data that we take as our reference standard, because we assume the clinical interview data is going to be more valid. We used that to then calculate the rate of false positives and false negatives when using screening tools, because there's always some of both. And we adjusted the prevalence estimates for both false positives and false negatives to try and get what we hope is a more accurate prevalence estimate. So in using our clinical interview data, these are the estimates we had. So for PTSD, it was around 40%. Nearly half of children met criteria for one or more anxiety disorders, around 20% for depression, and around just over a quarter for externalizing behavior problems. So we can see that these are sort of obviously significantly elevated beyond what we would see in other populations, and actually higher than we see in some other refugee populations. When we look at the whole sample, we looked at the prevalence at both baseline and follow-up. And one of the things I want to highlight on this, so we've reported both the raw prevalence estimate using the questionnaire measures and the locally validated cutoff, but then when we adjust that for the rate of false positives and false negatives, we find that those estimates decrease. So we can sort of appreciate that without adjusting, and the reason that decreases because we tend to see a much higher rate of false positives than false negatives. So it's really important to take that into account. At follow-up, we see similar prevalence rates. So they've, for PTSD, anxiety and depression, they tend to have decreased a little bit over time. And we see for any common mental disorder that over half of children are likely to meet criteria. We then looked at comorbidity, and this is data from the clinical interview, subsample with clinical interview data, but we see essentially the same pattern when we look at the whole sample. So what we see is that it's very rare for children to meet criteria for one disorder. They tend to meet criteria for multiple disorders. And commonly what we see is that children meet criteria for PTSD and then have other symptoms as well of anxiety, depression, and externalizing behavior problems. We think it probably makes sense to think of these as complex presentations or complex trauma presentations rather than distinct disorders. But this is the norm, that children meet criteria for multiple different problems. We looked at predictors of mental health problems. We used linear mixed effect models, and these were adjusted because subjects were clustered by settlement. Our outcomes were the total scores for each symptom scale, so that's PTSD, depression, anxiety, and externalizing behavior problems. We included as covariates child and caregiver age, child gender, and time since leaving Syria. And we also carried out a range of sensitivity analysis, including only those cases who had follow-up data, only those cases who had complete data, so no missing data at all, and also in subgroups that were defined by gender, age, and time since leaving Syria. And I'll just mention now that all those sensitivity analyses showed essentially the same pattern of results. So results were robust to those different types of analysis. When we look at predictors, so this is along the top, you see the 4 different outcome measures. The first predictor was time. So we see for depression, anxiety, and PTSD that symptoms tended to decrease over time. At follow-up, symptoms tended to be lower. For age, we saw small effects for depression, PTSD, and externalizing behavior problems. For gender, we saw the expected pattern that girls tend to have higher levels of anxiety symptoms and boys higher levels of externalizing problems. But what I really want to highlight here, what I think is really important, is that we found significant effects for all outcomes for the effect of war events. So the sort of past exposure to war events significantly predicts children's mental health symptoms, but the current environments, the perceived environment, is essentially sort of daily stressors. And we find for all outcomes other than anxiety that there's a much larger effect size there. It's an order of magnitude greater than the effect of the previous exposure to war events. This is a continuation of the same analysis, and this is just to show that we also found small but significant effects for child maltreatment and conflict between parent and child. So that those predicted children's mental health problems, and also caregiver mental health problems. So across all of children's outcomes, caregivers' mental health problems had small but significant effects on children's mental health problems. So to summarise, the burden of common mental health problems in refugee children is high, and in this population of children who are living in informal tented settlements, it's higher than we see even in other refugee populations. Comorbidity is common, or as I said, potentially maybe better to think about it, these sort of complex presentations where children have symptoms of multiple different problems. And this provides support for transdiagnostic approaches, such as transdiagnostic interventions. I've mentioned a paper that we published on this, where we sort of talk about these sort of more complex presentations in more detail. The relative importance of the quality of the environment, including daily stressors, highlights the significance of addressing social determinants of mental health. That seems to be the strongest predictor of children's current symptoms. Integrating mental health services with other services, such as schools or improved housing and so on, is important, but ultimately policy change that's aimed at reducing social inequities will be essential if we want to improve refugee children's mental health problems. Thank you very much.
Great, thanks a lot, Fiona. And just as a reminder, if anyone has questions, just to put them in the Q&A. I see there's already one question. So as people put in questions, the presenters will be able to respond to them, and then we may have some time later to discuss them. So the next presentation comes from Cassie Popham on the resilience in these children. So the previous presentation was mostly about the mental health, and obviously many children affected, but there's also some children that seem to be resilient. So Cassie's going to share about that. So feel free to share your slides. Thanks a lot.
Great, thank you. There we go. Hopefully you can see that. So thank you. As Michael said, my name is Cassie, and I'm a PhD student. And the focus of my PhD has been working on the BIOPATH data, looking at children's psychological responses to war and displacement, with a particular focus on psychological risks and resilience and predictors of those mental health outcomes. So today I'm going to focus on presenting my analysis from my first paper, which is trying to estimate how many children in the BIOPATH sample can be described as resilient, and what factors are associated with that. So as we've seen, Syrian refugee children are at increased risk of mental health problems. And according to the findings that Fiona just presented, the current sample may be at particularly increased risk. However, as Michael mentioned, there are a proportion of children in the BIOPATH sample and across refugee children in various populations that seem to show no evidence of psychological problems. These children could be described to be resilient if using the definition of having functioning that's better than expected, given the context of adversity. There's a whole range of predictors that have been associated with these outcomes from the individual levels, such as optimism or coping strategies, the family environment, such as the parent-child relationship, caregiver mental health, and so on. I've just included a figure here sort of illustrating some of the predictors that have been found in the literature. However, if we want to look at resilience, it's really important to look at multiple dimensions of functioning. As Fiona mentioned, there's often fairly high comorbidity, but there's also the chance that children could be doing well in one dimension and doing poorly in another. It's also important to consider the effects of war exposure about children's specific war experiences and what effects that have. So, for example, is a child doing better because of certain resilience factors that are helping them to adapt? Or are they doing better perhaps because they had slightly less severe exposure. So this leads me to the sort of two aims that I'm going to be talking about today, which was to estimate how many children in the BIOPATH sample could be described as resilient, and what differentiates these children from children who have been exposed to very similar war experiences but aren't doing so well. In order to look at this, I split the sample into two groups. A sample of low symptom children who might be described as resilient, and a sample of children with higher mental health symptoms. I then matched each low symptom child with a child with high symptoms, but who had similar patterns of war exposure, as well as similar age, gender, and a similar amount of time since living in Syrian. Then finally, I looked at what factors differentiated those two groups. So for this, I focused just on wave 1 of the BIOPATH sample, just baseline data. And I excluded any children who had reported no war events at all, as well as those who were missing any key pieces of data. This left me with 1,528 children before matching, about half of whom were girls, the majority of whom had their mothers participating as the primary caregiver. And on average, these children reportedly experience almost 10 different types of war events. So the majority were exposed to experiences such as witnessing explosions, but a significant proportion also were exposed to more direct interpersonal violence, such as witnessing torture. Given these fairly extreme experiences, I have considered that any child who is showing no or low symptoms of depression, PTSD, and externalizing behavioral problems to be functioning better than expected in the context of adversity. In other words, to be resilient. Just a quick note that I am focusing on these 3 outcomes and not anxiety due to the rate of false positives, particularly in the anxiety measure. Because I'm looking at categorizing individual children, I haven't been able to adjust for false positive rates in the same way as Fiona presented. So I'm just focusing on these 3 outcomes. So a child who was scoring below the clinical cutoffs for depression, PTSD, and externalizing problems was classed as low symptom or demonstrating resilience. However, if a child scored above any single cutoff, we consider them to be high symptom. They are at greater risk for having mental health problems. And in total, 295, so almost a fifth of the children met those low symptom or resilience criteria, which is a fairly small proportion. I found high symptom matches for every low symptom child. So you can see here that the groups were very well matched on the patterns of different war experiences that they reported, as well as on their age, gender, and time since living in Syria, as I mentioned before. I then looked at which factors differentiated these groups. And here is a list of all of those ones that were significant. As you can see, there's quite a range. In particular, self-esteem, optimism, and perceived social support were associated with an increased likelihood of belonging to the low symptom or resilient group. As you can see, there was a slightly higher number of factors that were associated with membership in the high symptom group, including environmental sensitivity, child maltreatment, and aspects of caregiver mental health. Most of these findings are to be expected, apart from the results for support seeking and problem-focused coping. These 2 types of coping strategies are often viewed as positive or adaptive coping strategies. But as you can see here, they're associated with high symptom group membership. Now, this could be a function of the context that children are in. So the sample is in a particularly challenging environment. It may be that more active coping strategies, such as problem-focused coping, can't actually help children deal with the problems they're facing, which could lead to more stress. Alternatively, it could be that children with more mental health problems have a greater need to try and use coping strategies. So the direction of effect could be in the opposite direction. So that is something that we need to look into further. Another important thing to consider is the interplay between predictors to understand how all of these different factors are actually impacting child mental health. So please forgive my slightly messy correlation plot. These were all the different predictors that I tested to see whether they're associated with resilience. What you can see here is that a lot of them are correlated with one another. And this can tell us a little bit about the processes within the environment and also maybe explain some of the results we found. So you might have noticed that Fiona presented that the refugee environment was a particularly important predictor of child mental health, whereas I didn't find it to be significant in this analysis. There are a couple of possible explanations for this. It may be that because I've got a fairly small subsample of the cohort, the sample I've chosen doesn't have enough variance in terms of the environmental quality to see any effects. However, the effects of the refugee environment are fairly inconsistent throughout the literature, and this could be because they have indirect effects on child mental health, perhaps via effects on the family. So according to Bronfenbrenner's ecological systems theory, wider aspects of the environment tend to impact the child via impacts on more proximal systems. So in this case, it may be that the refugee environment could impact the child via impacts on the caregiver. While I haven't tested this, we can see that looking at different mediations, so the refugee environment is associated with different aspects of caregiver mental health. Obviously, as I say, this can't tell us about mediation, but it could give us an indication that that's a pathway worth looking into. Similarly, we also see correlations between aspects of parenting and caregiver mental health, for example, and between different aspects of the social environment. So to summarize, 19.3% of the children in the sample met the low symptom or resilient criteria. This number might be higher due to the rate of false positives, as Fiona showed before, but nevertheless, this is a fairly small number. It emphasizes the importance of looking at multiple different dimensions of outcomes, and also suggests that this sample may particularly be in need of mental health support. I've identified several potentially important factors that predict outcomes. In particular, aspects of the social environment, parent-child relationship, caregiver mental health seem to be really important, but so too are individual factors such as self-esteem. As I mentioned, the effects of coping strategies, which were in the opposite direction to the expected, emphasizes the importance of looking at the directionality of effects in future work. And the correlations between the different predictors indicate the importance of taking the whole context into account and looking at the pathways to child mental health. Some of this I have looked at in the course of my PhD, but for the sake of time, I haven't been able to include here. So maybe we'll save that for the next webinar if and when that happens. So that is everything from me. Thank you very much. And obviously questions in the Q&A.
Thanks a lot, Cassie. That was great. Yes, so many of those findings that Cassie referred to are actually published. So we might send a follow-up to all the people that registered for the webinar with the references and links to those papers, which are open access. We will also make the recording of the webinar available. And thanks for those that posted questions. Feel free to post more. So we move to the third presentation, which is from Dr. Demelza-Smith, and she will focus more on PTSD and one of our biological measures.
Thank you. Yes, so yeah, I'm going to be sharing some of the biological findings from the BIOPATH cohort. This work has actually just been accepted for publication in Molecular Psychiatry. So if you want to read more about this work, you can check that out. So specifically, we're going to be looking at the relationships between war exposure, post-traumatic stress symptoms, and hair cortisol within our cohort. So why are we interested in cortisol in this context? It's been well established that adverse events like war exposure can increase the risk of poor mental health, as Fiona presented earlier in this session. However, we really want to better understand the biological mechanisms that link these findings. So a commonly investigated mechanism is the HPA axis, you can see on the right here. And this is the main stress axis within the body. It responds to psychological and physiological stresses, ultimately releasing cortisol, our main stress hormone, amongst other hormones as well. So altered functioning or dysregulation of this axis is commonly as following different forms of adversity, and also it's found alongside poor mental health. However, the exact phenotypes observed can vary quite considerably. Sometimes you see elevated cortisol, other times lowered. We sometimes see altered reactivity to acute stresses or potentially altered trajectories over time. So it's possible this is explained by the various different study designs, different populations, different adversities or mental health conditions studies. It may also have something to do with the time since the adversity, as there's an idea that following adversity, there's initial hypersecretion of cortisol, representing an overactivity of the HPA axis, which then leads into a longer-term hyposecretion. So people have investigating cortisol as a potential biomarker of adversities like war exposure and PTSD. And there is mixed evidence from cohorts of adults and adolescents that cortisol may be associated with these factors. However, as before, many of the findings, the exact findings do differ quite significantly. Other gaps in the current literature are that there are currently very few, if not no, studies looking at cortisol as a biomarker of war exposure or PTSD in children specifically. There are also, as was mentioned earlier in this session, very few studies looking at individuals that are still living in very adverse conditions like a refugee camp. Many have moved into a higher income country. And often many of these studies are quite small as well. So the main research questions we had for our study is firstly, whether hair cortisol as a measure of basal or average cortisol secretion is associated with war exposure, whether the hair cortisol is associated with PTSD symptoms in our war-exposed children and adolescents, and finally, whether hair cortisol mediates the relationship between these two factors. So as many of the other people in this session have described, we used both years of data that have been collected. So first of all, the PTSD symptoms, which is a self-reported scale taken from the children. We also looked at that. We also used their war exposure score specifically recorded at year 1. This was a combination of both the child and caregiver reports of the different types of war events that the child would have experienced. And as you can see here, we have quite a wide variation in responses for both the PTSD symptom score and the number of war events that the child experienced. We also collected a wide range of demographic, health and hair related variables, which are critical to include within such models, as many of these have been found to be associated with hair cortisol as well as mental health. So for example, within our cohorts, on average, the females tended to have higher hair cortisol than the males, and those that reported performing some kind of hair alteration, such as henna dyeing or chemical treatments, also exhibit higher hair cortisol. Finally, we were able to collect hair from the vast majority of our sample. We decided to collect hair in order to measure cortisol because it is quite easy to obtain from children in a humanitarian context. And it also negates the sort of temporal fluctuations in cortisol that you may find in other tissues such as blood. The hair samples were processed by our colleagues at Northwestern University, processed in the normal manner, and the extracts analysed for cortisol concentration using an immunoassay. And after removing a few outliers, we actually had quite a large sample remaining. We had 1,579 samples from our year 1 and 933 from our year 2. So on to our first question is whether hair cortisol is associated with war exposure. So here we can see plotted war exposure score against a batch corrected cortisol concentration from our hair. And as you can see, there is a weak but positive association between the two. We analysed the data using linear mixed models to incorporate both waves of our data, while controlling for the fact that we have repeated samples from the same child. Here I'm just going to present the main effects of war exposure on hair cortisol for simplicity. When we control for just batch, we do see a significant association between war exposure and hair cortisol. When we include a good number of covariates which are critical to include in such models, we actually don't see any significant relationship anymore. So we wanted to investigate a little bit further what it is that's confounding this relationship. And we found that actually hair age was having a great impact. Once we removed age from our models, we again saw a significant association between war exposure and hair cortisol. I would, however, like to point out that obviously these are quite weak effects. So for every additional war exposure or war-related event that the child experienced, that was only associated with around a 1% increase in hair cortisol. So we wanted to look a little bit closer at why age was compounding this relationship. So when we looked at here is the same sort of graph, but coloured by their age at interview. And we can see that actually the older children tended to have higher cortisol concentrations. They also tended to report a greater number of war-related events. So the association between age and cortisol is somewhat backed up by the literature, although the findings are incredibly mixed. On the other hand, the association between age and war exposure is perhaps understandable, given that children that were older during the interview would have been more likely to be, would have been older during the Civil War itself, and possibly able to just experience a greater number of war-related events because they were away from home and not necessarily being shielded by their family. We wanted to take a closer look at this. So we stratified our sample by their age at the end of war exposure, so their age at the time of leaving Syria, and repeated the same analysis. So we looked at the association between war exposure score and hair cortisol for these 5 age groups. And we can see for the first 4 age groups, we don't actually see much in the way of an association between war exposure and hair cortisol, although obviously you can see the shifts in war exposure score and hair cortisol as you move through the ages. However, in our oldest age group on the right-hand side, who are aged at least 12 at the end of their war exposure, we do still see a positive association between war exposure and hair cortisol, even when controlling for age at the interview. This kind of suggests that perhaps this age group is slightly more sensitive to the effects of age and have somehow sort of biologically embedded the adversity more than the other age groups. We also wanted to look at how hair cortisol was associated with the time since leaving Syria, or the time since their exposure to war ended. Here is the time since leaving Syria plotted against hair cortisol. And while the effects are, again, very, very small, we do see a very slight decrease in hair cortisol as in the children who left Syria a longer time ago compared to those that left within the previous 12 months. We wanted to see, therefore, whether time since leaving Syria or time since their exposure interacted with the war exposure. Therefore, we did another stratified analysis time since they left Syria, again looked at the association between war exposure and hair cortisol. However, we don't really see any particular evidence for an interaction effect here. All of the associations seem pretty similar across the two, and there isn't really much going on here. So therefore, we can't really say whether we have any evidence for this sort of initial hypersecretion of cortisol moving into hyposecretion. So on to our second question, which is whether hair cortisol was associated with PTSD symptoms. Again, here we have plotted PTSD symptoms against batch-corrected log cortisol. And again, there is a positive but weak association between the two. Again, we analyze the data using a linear mixed model method. And here I'll be presenting the main effects for PTSD symptoms on hair cortisol. So again, when just controlling for the analysis batch, we do see a significant association between the two. And even when we add in these additional covariates into our model, we do still see a significant association. Just to be able to compare it back to our findings within war exposure, we did repeat the same analysis, removing age at interview. And again, it does seem that perhaps age at interview is somewhat confounding this relationship. However, as before, these associations are really quite weak. So for each additional PTSD symptom, that the child reports, on average, there was about a 0.5% increase in hair cortisol. So on to our third question, which is whether hair cortisol mediates the relationship between war exposure and PTSD symptoms. So we wanted to test two different mediation models here. The first on the left, asking whether cortisol acts as a mediator between war exposure and PTSD symptoms. And on the right, whether PTSD actually acts as the mediator between war and hair cortisol. Our first mediation model, it seemed like cortisol is a significant partial mediator, but mediating a very small proportion of the relationship here. Our second mediation model with PTSD as the mediator also showed significant mediation, indicating a slightly larger proportion of mediation. Therefore, it does seem that these three variables are related in some way. However, we cannot say in which manner they are related. So on to the conclusions from this project. We found that hair cortisol is indeed weakly associated with war exposure, but this finding is confounded by age at the time of their interview. So future work will need to be done to disentangle this sort of relationship of age from war exposure. It seems like early adolescence may be a particularly sensitive time period to adversities like war, due to seeing the stronger relationship within children who are at least 12 at the end of their war exposure. However, as this was an unplanned post-hoc test, this will need to be replicated in an additional sample. We also found that hair cortisol is weakly associated with PTSD symptoms, and that these three factors, war exposure, PTSD, and cortisol, are linked in some manner, but the exact mediation is not entirely clear. However, finally, I would like to just highlight that demographic and technical variables which were included within these analyses were far more strongly associated with hair cortisol than either war exposure or PTSD. So just as an illustration, if we look at children who scored the minimum all the way up to the maximum war exposure, those only exhibit around a 39% increase in hair cortisol from the two ends of the scale. On the other, in comparison, females exhibited about 63% higher hair cortisol compared to males. And these sort of differences were seen in many other of the technical or demographic variables used. This sort of suggests that not only are these really important key things to include in such models, but also it suggests that hair cortisol may not act as a particularly effective biomarker for war exposure or PTSD. So thank you all for listening, and thank you for all of my colleagues.
Thank you very much, Demelsa. Very interesting findings, particularly that it seems to be young adolescents where we do find a biological impact or biological signature of war exposure, which just shows how important it is to consider development of these children and to look at different ages. Great, so we'll have now Professor Eli Karam sharing some thoughts to discuss these findings. So Eli, feel free to start sharing your slides.
Okay.
And feel free to everyone else to add questions to the Q&A, because we'll have some minutes after that to discuss. So any urgent questions that you would like us to discuss, please let us know in the Q&A.
Okay. Is this clear?
Yeah, we don't see the slides yet.
Okay. Let me share screen here. What's happening here? Sorry.
Otherwise, I can share your slides on my computer.
Could you please? Yeah.
Yes, let's do that.
Is that it?
Are
they on now?
They are on now. Yes.
Yeah.
Yeah, you can start.
Okay. Thank you. I would like to first thank a lot the Queen Mary team for the fantastic work that I've been associated with, and DRAC, our group in Lebanon, have been associated with for many years. And it looks easy from the outside, but from the inside, it was really still is a fantastic endeavor, especially to undertake a longitudinal study. And now the uniqueness of this BIOPATH study is that really the wars are the, if you want to, natural experiments where we can study the effect of trauma, large scale, very large scale on humans. And to add to that, to take samples of children, adolescents, and their families in very difficult circumstances, where we were not sure whether we're going to be able to continue the studies at any time because of the ongoing conflicts, internal conflicts that were happening even in these camps, and the looming problems that could happen in Lebanon because of these camps. So, and to add to that, a prospective study, this was really something very, very important. And as you have witnessed already, gathered from the previous presentation, to look at the very, very intricate nature of environmental, biological, and personal journeys of each child adolescent was something that really very happy that could be done. Now, clinically, one aspect of it is that it has many implications, this BIOPATH study, and we're just sharing some of the findings so far. Many are still to come. And who gets affected, you know? And not everybody gets affected, fine, but why not? And what are the individual characteristics? And lots of work has been done on that, and follow up prospectively, not just retrospectively, who has been able to escape the scars of war and the environment that these children have been exposed to. Another thing is just heard about the biological reflections of trauma. While one can read papers that can state easily that cortisol is down or whatever after a while, and that makes you more susceptible to experience more trauma or more reactions to trauma. The reality is at times more difficult and might change, as we have heard, from age to age, which is something that is very eye-opening. And interestingly, what would be the long-term sequelae of trauma? Are we marked for good? Is it going to be following us up, as some people have suggested? And sorry. The trauma itself, when we look at it, it's very simple, straightforward. War, exposure, but many things occur around this trauma. The environment you're living in after the trauma, including your personal immediate environment, and very important to keep in mind. And frequently over the years with our research, we have seen that people have neglected the effect of environment and focused on more on the trauma itself. And more we're finding that the environment is at least as important, if not important. And so to see that coming out in research is really very interesting and reassuring that this team at Queen Mary has been able to look at all aspects of intricate aspects of the environment and the individual factors, and at the same time look at different in different ways, creatively, at how these unfortunate kids have been living in environments and in settings that might or might not be helpful for them. And I mean, this is something very important. So it's really a very thrilling scientific journey for those of you who love science. The meticulous attention to details is unbelievable. And we have not mentioned the internal struggles that we faced and how to get this study to take off in Lebanon, and to collect genetic material, etc., and to ship it abroad. It's not something that is straightforward. And so and to be to face practical issues, how to get these kids to get treated. It's not straightforward. We have not presented that this time, but you can imagine the attention that was given to these kids. It's part of any humanitarian research, is not as easy. And I cannot but stress that the environment is equally important, if not more important, than the trauma that we're studying. And thank you very much. Thank you.
Thanks a lot, Eli. So we have just a few minutes left for any questions. Should there be any questions? There was one question that I think is quite helpful, and asking about how can we use these findings to help refugee children. Well, this was the goal from the first moment of this study, is to find out what helps children to be resilient, and what can we learn from resilient children to help those that did not prove to be resilient. What are the factors that are important to promote in those children? So this is still the early stages in that we are finding out things and we are publishing these findings, and then the next step is how to apply these findings in practice. But our hope is that our research results would inform intervention, would also inform the need, what is actually the actual need for the treatment of mental health problems among these children. We did not expect to find those high rates of mental health problems as we did. And we expected to find more children that would meet our criteria for resilience. And one of the reasons why the proportion of children that are resilient is a bit smaller in our study is because we considered multiple outcomes at the same time, whereas other studies may only focus on one outcome like PTSD. And as we did in our study, would find that quite a few seem to be resilient when actually they did not develop PTSD, but they might have developed something else. So further questions, we'll have to end soon. The mental health of the parents of the resilient children. This is a two-way relationship. This is exactly what we find. We have a paper that will come out looking, that actually already is out, that looked specifically at the bidirectional relationship between parental mental health and child mental health. Very important to look at. It's very important to look at the development over time. And we'll have to end here, but I'd like to just say that please stay in touch with us. We'll send a follow-up email to all those that registered. We will make a recording of the webinar available. Hopefully, we will be able to share some of the slides as well. And please feel free to get back to us if you would like to be invited for the next webinar, which will probably be sometime in March or April next year. We are also in the process of setting up a website where we will share these webinars. We will share all the publications and the information from this research. We want to make sure that as many people as possible will benefit from these research findings. Great. So I'll respond to some of those questions in the Q&A. But for now, I'd just like to thank all the speakers and all the presenters. Everything that was presented today has already been published or will be published very soon. So hopefully you will be able to see some of those findings in print very soon. So thanks a lot for taking time to attend this webinar, and we hope to see you in the next one. All the best. Bye-bye.